Welcome to the Personalized Immunotherapy Research Lab or PIRL YouTube channel. I'm Stephen Vensko from the Vincent Lab at the University of North Carolina at Chapel Hill, and I'll be your guide today. In this video, we're introducing PIRL's neoantigen workflow lens, an acronym for Landscape of Effective Neoantigen Software. We'll cover three main topics. We'll start off by discussing what lens is. Then we'll explain why we built lens. And finally, we'll cover what's required in order to run lens on your own samples. We'll go over installation and running lens in a future video, but for now, let's focus on understanding lens. Lens is an end-to-end workflow for predicting both neoantigens and tumor-associated antigens. Lens starts with FASTQ files and ends with a list of predicted peptide MHCs, also known as PMHCs, that may be biologically interesting or clinically relevant. These PMHCs can originate from single nucleotide variants, insertions, deletions, splice variants, fusions, viral or retroviral elements, or overexpressed self-antigens. Along the way, lens also generates a number of intermediate outputs that may be useful for downstream analysis. The workflow is managed using RAFT, our Nextflow-based analysis framework. If you're not familiar with RAFT, you can check out the video linked in the upper right or browse our channel for videos related to RAFT. Now, let's talk about why we built lens. It's true, the neoantigen computational prediction space already includes many workflows. In fact, when we published the original lens manuscript, we compared it to 26 other similar workflows. Clearly, there are several available, so why create another one? We noticed some common issues among existing workflows. Many were hard-coded with specific tools or tool versions, making them rigid and difficult to customize. Even more concerning, many had not been updated in years. The last time we checked, the average time since the last update was over 600 days. We built lens to address those concerns. Specifically, we wanted a workflow that was modular, extensible, and maintainable. Let's break that down. Modularity. Lens is built upon RAFT, which introduces modular architecture. That means users can swap in different tools, for example, using their preferred aligner, variant caller, or HLA typing method without having to rewrite the entire pipeline. Extensibility. Lens is designed to grow. Right now, it supports short-read Illumina sequencing, but future updates will include support for long-read and single-cell data, among other data types. Maintainability. The modules in Lens follow a process definition standard that helps us rapidly update modules for new tool version and features. This keeps the workflow up to date and flexible as methods evolve. We'll go into more detail about how Lens works in future videos, but if you're curious, you can find more information at uselens.io. If you've made it to this point, you're probably wondering what it takes to run Lens on your own data. To get started, you'll need a few key inputs. You'll need a normal DNA sample, a tumor DNA sample, and a tumor RNA sample, all from the same patient. You'll also need a tissue-matched normal RNA sample. Tissue-matched normal RNA samples can be sourced from GTEx or other similar sources. Finally, Lens requires a manifest file that defines the relationships between datasets, patients, and samples. We'll walk through how to create a manifest file in another video, but in the meantime, you can find guidance and examples at uselens.io. And that wraps up our introduction to Lens. Thanks so much for watching. If you found this video helpful, don't forget to like, subscribe, and hit the notification bell so you don't miss future uploads from PIRL. If you have questions or want us to cover a specific Lens feature in detail, then please drop a comment below. We'd love to hear from you. Until next time, take care and happy analyzing.